Back to Biology 121 Online. This is our fourth video. We're going to be talking today about photosynthesis and particularly problems and solutions that plants face in carbon fixation. So you may recall that carbon fixation is sometimes called the light-independent reactions in photosynthesis. It's what happens after all of the solar energy gets converted into chemical energy in the form of ATP and NADPH. We recalled last time that plants breathe in carbon dioxide, if you want to put it that way, and breathe out oxygen. The difference between CO2 and O2 is that carbon atom. The carbon stays inside the plant, and that's why we call it carbon fixing. The carbon is affixed to the plant. That happens through what's called the Calvin cycle. Again, this is all review from video 3. It's like the Krebs cycle, but in reverse. And just like in the Krebs cycle, in cellular respiration, there are 2 turns of the Calvin cycle for every sugar molecule that's produced. You'll see at the bottom here, we're looking at an output of a half sugar, half of a molecule of glucose. Stored energy there, and then those half molecules get separately assembled into sugar. And the Calvin cycle needs 3 things to work. It needs carbon dioxide from the air, which gets absorbed by the leaves, that was at the top of the cycle. It needs water, which comes up from the ground, absorbed by the roots. And it needs rubisco, which is the enzyme that the cycle produces and reproduces at the end of the cycle. That's why there's that second half of the circle is regenerating the rubisco that binds the carbon dioxide from the air in the first place. So remember that photosynthesis happens within the leaves of the plant, and the leaves of the plant are usually up high so that they can catch the sunlight. So how do plants pump water up from the ground up to the leaves? Sorry, that says from the group up to the leaves. It should say from the ground up to the leaves. And obviously the water isn't just going to levitate up through the plant under any normal process. The plant doesn't have any muscular tissue like a heart to pump that water through the plant. So what is it that allows the water to end up going upwards against the force of gravity? And the answer is a process called evapotranspiration. Take a second to wrap your head around that word, evapotranspiration. Plants are like big drinking straws. See, they have these hollow tubes inside, and that allows water to evaporate from the leaves using the same holes that bring in CO2, and that evaporation process draws more water up, like sucking it through a straw. So evapotranspiration is a fancy term for evaporation that transpires through the plant. So it's that force of evaporation drawing water out of the leaves of the plant that causes more water to enter through the roots of the plant. So, we talked last time about what plants do with all this sugar they're producing through their carbon fixation process. They form either cellulose, which is the woody part of the plant, or starch, which is the nutritious part of a grain. And here's a question, though. So we just talked a little bit about how water is coming in through the bottom of a plant and going up and evaporating out through the leaves. Well, if the air around a plant is hot and dry, the plant can very quickly lose all of the water that's available to it by evapotranspiration. So, as we know, in hot, dry conditions, plants can end up draining the soil around them of any water, and then the plant is going to die because it doesn't have access to the water. It can't participate in this Calvin cycle anymore, Krebs cycle anymore, Calvin cycle. So when the air around a plant is hot and dry, plants will close off some of their stomata. Stomata is just a Greek term for hole. So that's the pores in the plant leaves that allow carbon dioxide to enter and allow water to evaporate out. So by closing some of those holes, closing some of those pores in the leaves, they can slow down the loss of water. But that also slows down their ability to absorb CO2 because they use the same pores to exhale water, as it were, and to inhale carbon dioxide. So shutting down one of them shuts down the other. Without the carbon dioxide, there's no carbon fixation, there's no sugar production. So when the air around a plant is hot and dry, plants will take in less CO2. As it happens, the enzyme that binds carbon dioxide, called rubisco, can run the cycle backwards, binding oxygen and releasing carbon dioxide. And this is counterproductive in terms of carbon fixation. It actually causes the plant to give up some of the carbon that it would otherwise use to form its body. So some plants, some species of plants that are not well adapted to hot, dry conditions can lose up to 50% of their carbon fixing ability to this process that's called photorespiration. So let's take a quick look at photorespiration and photosynthesis. Photorespiration is called photorespiration because it's absorbing oxygen and releasing carbon dioxide, just like any other respiratory process. That's the exact opposite of what photosynthesis is supposed to be doing. So on the left-hand side, we have this negative, lossy photorespiration. And on the right-hand side of this picture, we have the carbon-fixing, sugar-producing photosynthesis. That rubisco can run the process either direction. And so the real trick then is how do you make sure that you are getting enough carbon dioxide to make sure that your rubisco is preferring photosynthesis while also not losing too much water. So some plants are better suited to hot, dry climate than others. And here's some reasons why. There's 3 different ways that plants can process carbon dioxide. The way that we've talked about so far with the Calvin cycle is 3 carbon dioxide molecules get bound to rubisco and immediately enter the Calvin cycle. Because it's based off of 3 carbon dioxide molecules at a time, we call this a C3 pathway. C for carbon, 3 because there's 3 of them. Alternatively, some plants use 4 carbon dioxide molecules that get fused together into a different carboniferous molecule, and those are assembled in an outer cell that has contact with the air outside of the leaf. But then those 4 carbon molecules are handed off to an inner cell where the Calvin cycle actually occurs. So there's no rubisco out in the outer area of the cell, which means if there's an abundance of oxygen and not as much carbon dioxide around the plant, it doesn't matter because where that rubisco is starting the Calvin cycle is further down inside the plant. We'll take a look at a drawing of that in just a second. There's also a third version which sounds similar. You get 4 carbon dioxide molecules that are fused together, but in this kind of plant, that fusion happens at night. So the carbon dioxide gets absorbed into the leaves overnight, and then the Calvin cycle occurs during the day when the ATP production and NADPH production is happening because of the light-dependent reactions. So let's just take a quick look at each of those 3 pathways. We've talked about the Calvin cycle in our basic C3 pathway, where 3 carbon dioxide molecules come in, the rubisco binds those together, and then the Calvin cycle turns those into sugars. This occurs in what are called mesophyll cells. So these are cells that are happening, that are like the outer cells of the leaf. So mesophyll means at the exterior, the boundary between the leaf and its environment. In a C4 pathway, the mesophyll cell doesn't participate in the Calvin cycle. You see over on the right-hand side, the mesophyll cell is that narrower, thinner cell that is absorbing carbon dioxide and binding it together using a different red lozenged enzyme into a 4 carbon, see the 4 little gray dots there, a 4 carbon molecule called oxaloacetate. That oxaloacetate then gets changed into another 4 carbon molecule called malate. That malate is then transferred into a lower down cell called a bundle sheath cell, where 3 malate molecules will give up 3 carbon dioxides, and then you have the normal Calvin cycle starting down there. Each malate, then having given up a carbon dioxide, is degraded into our old friend pyruvate. So this is an example of plant cells using the same molecule in two very different processes. So it's a little bit of an efficiency. And then it takes a little bit of energy once that pyruvate comes back out into the mesophyll cell to regenerate that pyruvate into a 3-carbon molecule that's going to get a CO2 bound to it to become oxaloacetate, repeating the upper part of the cycle. So here we have two separate cycles. We have the oxaloacetate, malate, pyruvate, PEP cycle, and then we have the Calvin cycle below that. And that's literally happening further down inside the leaf. Lastly, there's the CAM pathway. In the CAM pathway, we also again have that same secondary enzyme, the PEP carboxylase, which is forming carbon dioxide together with PEP into a 4-carbon oxaloacetate, which then gets changed into a malate, also a 4-carbon. In some plants, there are other organic compounds with 4 carbons involved in them, other than malate. And all of that happens at night when it's not hot and dry, when the plant can afford to leave the stoma open and draw in all of that carbon dioxide. Then, during the daytime, they close up the stomata, so there's no more carbon dioxide coming in, but there's this stored carbon dioxide that then gets processed in the Calvin cycle as the energy from the light-dependent reactions become available. So there's 3 separate pathways. The C3 pathway is the simplest. The C4 pathway only really happens in plants that are adapted to hot, dry conditions because it's a solution to the problem of over-evaporation. And the CAM pathway, which is a separate solution to that same problem that's related. It uses a lot of the same machinery, a lot of the same molecules in C4 versus CAM, but they're 2 slightly different ways of solving that same problem of how do you get enough carbon dioxide to ensure that your Calvin cycle is going to run without having too much water leaving the plant through those same pores. So I look forward to some discussion around these concepts. I think this one's a little bit tricky to wrap your head around, so please do, if you're able to, join us for the discussion or drop in and add some questions to our discussion document.